husband is handicapped because what it says is that you've been out, you've thought about things, you've been working, whatever, and you're bringing experience. We were talking about, I don't give somebody, I don't give somebody science if they aren't willing to work. You could be the most brilliant person in the world, but if you can't focus or you don't have any interest in working hard, my students, one of my students used to joke that she was my workaholic in training.
It's 3:30.
Yeah. And get started.
Okay.
Absolutely. Oh my God.
Yeah, we got a good crowd here today to learn about our local springs and slime.
Okay.
And it's with pleasure that I introduce Jim Garey. He's a professor in the CMMM or whatever.
CMMB.
CMMB, yeah. Cell and molecular and microbiology.
It's up there.
Oh, it's there, yeah. I don't have to say it. Jim had earned his PhD from UT Austin, University of Texas at Austin. Yeah.
Hook 'em horns.
And he started his career at Duquesne, is that what
Yeah.
you said?
Duquesne University.
At Duquesne University, and he came to USF in 1997 as a professor and has been here ever since. He he's into administration as well. He was chair of the department for a number of years. How many years?
12.
12, yeah. And now he's president of the Faculty Senate. And are you still the faculty representative on the Board of Governors?
Board of Trustees,
Board
yes.
of Trustees, I mean, not the Board of Governors, Board of Trustees, the USF Board of Trustees. So if you have important things that you need to know about, you can.
If you're faculty here, you got an email from me yesterday.
Yeah, exactly. The faculty here know we got an email
Vote
from
yes.
yesterday. I've known Jim for a number of years, partly because we both serve on the USF Diving Control Board. In fact, I was chair for many years, and then he took over as chair.
She made me.
And then we actually got to go on 2 field research expeditions to Papua New Guinea and dive on thermal springs that came out at about 6-7 meter depth and very fascinating study. And then somewhere along, and about that time, he really got into caves and slime, and so he went from being one of the premier invertebrate biologists to studying slime.
Sure. Okay.
Thanks,
Thank
Jim.
you. I'm going to turn this down
Oh yeah.
and see if I haven't lost this. Make sure it's on. Terrific. So thanks for having me, and thanks for inviting me, Pam. I have 2 students here to keep me honest, Bobby Sharping over there and Vicki Fraser, an undergraduate over there. So I can't make up stuff. I have to tell you what actually happens. So I'm going to be talking about some of the karst features we've been studying along the coast here, and it's been going on since we've published our first paper in this area in 2005. What Pamela was just referring to was before then, I worked in molecular phylogenetics of metazoa as a whole, and that's where I kind of made my original splash, I guess. But this stuff nobody cares about as much, but it's fun. It seems to be a lot more fun for me to do. So I'm going to talk first about why we do this work, and I want to talk about training, and this is mostly for the students in the audience. So I've never taken a course in microbiology, I've never taken a course in ecology, and I've certainly never taken a course in geochemistry, hydrology, or even geology, and yet that's all of what I'm doing now. So life, you go through and you learn continually. So that's, to me, a really important thing. I'm going to talk a little bit about, a little bit about the methods we use, and then I'm going to tell you 3 short stories. The first one is going to be about double keyhole, which is a tidally influenced coastal spring that creates an estuary that nobody even knew was there. So for springs, if you've lived in Tampa or been around Tampa, you know that that's in Tampa. It's a salt-influenced urban spring. And then I'm going to talk about Hospital Hole, which is a stratified submerged sink that lies underneath the Weeki Wachee River. So why is this work important? I think I'm preaching to the choir here, but generally Florida relies almost entirely on karst aquifers for water. Karst is prevalent all over the place. In fact, somebody was showing me a slide that showed all the karst in the world, and it's about 20% of the landmass. And not a whole lot of people approach karst the way we do. We go in it. And most hydrologists, they look at wells and they take samples out of wells, or they go to the spring head and pull a little bit of water out of the spring outflow and study that. But we actually go in it. And think about a water tap. So you go into the kitchen and you fill up a glass with water. You could analyze that water all day long, but you would probably never figure out the pathway that water took to get to your faucet, right? You wouldn't have known where the water was pumped from originally, all the processing it went through, all the plumbing it went through to get there. So when we go in, we see a little bit more about that water, and we see how much more complicated these springs are once you look at them as a whole. And each karst feature we look at is pretty unique. And you really can't make too many assumptions from one to the next. We're finding out there are some similarities, but we have to always look anew at everything we look at. And Karst provides lots of ecosystem services that I don't think I need to talk about now. So we use a lot of different methods. The first one is scuba diving. We go in the sinks and we can do direct observations and direct sampling. We use some equipment, these acoustic Doppler velocimeters and these hydrolab data sondes. We can deploy underwater and leave them there for several months, and they can record continuously. So this is Isabella Spring. It's a coastal spring in Hudson, and this is down about 60 feet. This is underwater, and this is like a 5-gallon bucket, like a Home Depot type bucket. And there's cement in it and a hole in the cement, and this is an acoustic Doppler velocimeter that measures current flow and direction. And this is the hydrolab data sonde that collects pH and all that kind of stuff in depth as well. And we do a lot of chemical analysis. We don't have a lot of money, so we use the Hatch spectrophotometric things to look at the nutrients. But we also do mass spec to look at stable isotopes. And then we do a lot of molecular biology analysis of DNA. And it's interesting, we use this Primer E software, which was designed for benthic community analysis. And yet we use it for microbial communities, and it turns out you can just drop in DNA sequencing results right into that program and use it as if they were direct observations of organisms. So it works out really well. To process all the sequencing that we do, we use this freeware because we're poor. So we use this Mother software. It's a public domain software package to analyze Illumina sequencing for microbial ecologists. So we can use all these tools to get a comprehensive view of what's going on in the spring. And we focus on microbial communities. Remember, I'm an invertebrate guy. I'd much rather be looking at the invertebrates. But over the years, I finally came to the realization that microbiologists are born with, apparently, and that is that the microfauna really determine what really goes on in a system. So they are the base of the pyramid, kind of, in terms of the ecological function of the water column and the sediment, at least in these springs. But they're invisible to most people, whereas macrofauna and flora, everybody notices if a bunch of fish die or if there's a big algal bloom. But we think we can predict those kinds of things by looking at the rapid changes that can happen to the microbial community. So here's the first short story. And this is Damian Menning, a former student. He's now at USGS in Anchorage. He wanted to get as far away from my lab as he possibly could, apparently. No, we're still good friends. And this is double keyhole, and this is a pond, and over in this area behind us is a sinkhole under the pond. And you drop down right about there and go down about 25 feet, and there's an opening, and that opening goes under the ground and it's a spring. So here's kind of a map of it. So this is an overview. There is an active limestone quarry here, but the spring comes out here and it discharges directly into that pond, and that pond is directly connected over here, and it actually creates this estuary. So double keyhole spring is a magnitude 2 spring. And this looks like this might be a river or something, but this was an old canal that was built, I think, in the 50s or something, and it's not even complete. It's been filled in various places. So this has never been an outflow of water particularly. So the water that's created this estuary here is this spring. And so we study what's going on in the conduit itself, what's going on in the pond, and then 2 sites, a near estuary site near to the pond and one further out from the pond, to get an idea of what's going on here. So this is what it's like working in the spring. So there's a lot of current sometimes, and there's a lot of microbial film on the walls and in the sediment. And so the water, when it's really flowing, it stirs up all this stuff. This isn't us being sloppy divers. This is all the crap that just comes out on a really busy day in there. So Damien is only about 3 feet away from me. There's his hands, and he's holding a 50-watt light in his hand. So that's kind of the conditions to work there. And again, this is some of our instrumentation that we've deployed. So it varies, but it's usually about magnitude 2 spring. It discharges from negative 1,500 liters per second to about 2,600 liters per second, and we've measured it ourselves, so we know that's true. It's brackish water, usually somewhere between 10 and 20 salinity in the conduit. At high tide, during dry conditions, that spring will reverse flow. So Gulf water or estuary water will go into the conduit. But that only happens about an hour a day, only when the aquifer level's low. The other 23 hours a day, it's flowing outward, bringing mixing zone water from deep within. And you saw all the stuff in the water, so that's nutrients that it expels out into the spring, and we think that creates, helps support the juvenile game fish. And this is down near where the cave entrance is, and I'm videoing up, and those are all the juvenile game fish that we see there all the time, hanging out. So our questions: how does a coastal karst spring function? Nobody ever published anything about one that we could find. Where does that water come from? We had our theories, and I can tell you that our theories were completely wrong, which makes me feel good that we're doing real science. What is the system like, and what drives change in the system? And those are the main things. So we did 3 years of field work, and we were sampling quarterly. So this is kind of a key piece of data. So this is a lot of data, but this part here, this is the Acoustic Doppler Velocimeter data. And here's our collection dates that we collected discrete samples, but we left the instrumentation in, and there are some gaps in it, but you still get a pretty good idea what's going on. This red mark here is when Tropical Storm Debbie came into the Tampa area. It stalled and sat over us for 3 days, dropped a huge amount of water. So if you look at rainfall here, over that period of time, here's Tropical Storm Debbie. So here's a dry season, wet season with Tropical Storm Debbie, another dry season, another wet season, etc. You look at the flow, here's 0, and this is flow outward, so positive flow. And before Debbie came along, it was usually around 1,000 liters per second. But notice it would drop below 0, typically once a day, but not every day, depending on the aquifer level. And then it was getting kind of crazy here. I guess we were having some rainfall come in. But then when Tropical Storm Debbie hits, we get this huge spike. And we actually had discharge up around the 2,700 liters per second maximum that we saw. But notice that it did decrease, but over the rest of the study, which goes from June 2012 all the way to January 2014, it never really completely dropped down to the pre-Debbie levels. It always stayed above that. So Damien went and looked at some of the nearby well data, and here's the hydraulic head in 4 different wells over time, and these are recorded by either SwiftMud or USGS, I suppose. And what you can see here is these 2 wells didn't really change much. They didn't recharge very much when Debbie came through, but these 2 did. So these went from being fairly low to really high. One of them stayed high all the way through the study. The other one dropped back down after about 6 months, dropped back down to the pre-level. But we think the reason this is sustained is because this particular part of the aquifer was chock full, I guess, of water, and has maintained that at least for the rest of our study period. So that's what we saw happen. So we looked at the geochemistry, we looked at nutrients extensively. We found only minor changes over the study period, and we were looking at things like sulfate, nitrate, ammonia, phosphate, things like that. And we did find variations in salinity, pH, and dissolved oxygen. But again, we didn't see anything really dramatic, so I'm not going to go through all that data because it just, otherwise I couldn't tell you the three stories, short stories. So when you go into the cave, I'm showing you these pictures. These are actually have something to do with Vicki Fraser's carbonate dissolution experiments, but I just want to show you this because this is a PVC pipe framework we put in, and I took that picture right after we put it in. You can see how nice and white and shiny the PVC is. Here's an older experiment that's been sitting in there, in this case for 2.5 years. But all this biofilm that grows on this PVC pipe would grow on this. So the very next time we went in here, this thing within a month looked just like this one. So you get this really fast growing biofilm in double keyhole. And we looked at the water column, we looked at archaea, bacteria, microbial eukaryotes, and we looked in the sediment as well. And we think that most of the microbial community you find in the water column is biofilm that's come off the walls and has mixed in with the water column. So this kind of summarizes some of our data. So just to orient you, up here is the discharge from the spring over time. So here's that same time period, same sampling times as Tropical Storm Debbie. This is the water discharge, daily discharge from the spring that we measured. And these are, you start at the spring, you go to the pond, you go to the nearby estuary site, and then the far estuary site. And the green here is archaea, microbial abundance done by qPCR. This is bacterial microbial abundance done by qPCR, and this is eukaryote microbial abundance done by qPCR, all either 16S or 18S. And what you see here is in terms of abundance, it builds up. And in some places, especially the pond and estuary 1, it's pretty high. And then right after Tropical Storm Debbie goes by, it drops way, way, way down. So you can just imagine like a big toilet flushing. All that water came out, and it just carried all the biofilm out with it. And so we saw a lot less of it in the water column. And you go to species richness. This we did by LHPCR. We didn't do sequencing then. We were just doing fragment analysis. But still, it gives us an idea of how many different species of microbes there are present. And if you see that, you see the opposite happen. So here's again archaea, bacteria, microbial eukaryotes. And what you see there is kind of moderate amounts in general, but then right after Debbie goes through, you see a quick increase in species richness. And so what's happening is you flush out most of the bacteria or most of the microbes, so that causes abundance to drop, but then you've got wide open new niches for different bacteria to settle, and when they're resettling it, you're going to get a higher diversity initially, and eventually they'll start competing with each other and it'll drop back down, which you can see that when you go into further dates. But the main peak was right in that first 3 months after Tropical Storm Debbie went through. This was for the water column. We showed the same thing with the sediment. I just don't have time to show you all of that. So here's what the system actually looks like. So here's the pond, and here's the estuary as you go away from the pond. If you go down there, you're actually going out toward the Gulf. The conduit doesn't run in toward shore, it runs out toward the Gulf. And this X is about where we collected most of our samples. We found that if you found water here, collected water here, here, and here the same day, they were exactly identical in everything, because water that's here ends up here within 5 or 10 minutes anyway. So the water's flowing through there at a pretty good clip. In terms of those reversals, we placed some instrumentation here, and we know that those reversals never reached this far in, and this was at 800 feet. So that's why we know that when the spring reverses, it doesn't flow past the first 800 feet or so. And as you go further back in the conduit, you reach this big room, and down at the bottom of the room is where the water is coming from. And so we hypothesize that there's a inlet into this system out here in the coastal aquifer. So imagine you have aquifer level here determining where the fresh water is coming, out this way, and then you have sea level, tides, and all those pushing water in this way. And so anytime you have drought or less rain, this freshwater, saltwater boundary is going to move inland, but when you have a lot of rain, then it's going to move outward into the Gulf. And so imagine you got a fixed intake pipe here, and you're moving this mixing zone back and forth across here, and that's why we get slightly small in salinity. As this boundary moves out that way, it's going to get a little less salty. As if the boundary moves in, it's going to get a little more salty. So that's kind of the general idea about how this probably works. So the primary drivers of the change that we were finding, biological change within the estuary, is really the amount of water that it's discharging. And we found that estimated abundance varies inversely, while species richness varies directly with aquifer discharge. And really, the communities change more on a dry season, wet season pattern than a regular seasonal pattern, which is what you'd expect. And again, we're pretty sure that the biofilm on all those nutrients discharged, we see, though, that same biofilm inside this conduit, you see it outside covering everything out in the estuary. You see a brown film on everything. So we're pretty sure those nutrients are what is driving the food web out there. So we published this. The first paper came out in Journal of Hydrology a few years ago, and the biological paper came out. This says 2018, but it actually was published electronically last summer. So that's that story. And now I want to talk about another story. How many of you know about Sulphur Springs, have heard of it? A number of you have. So it's right in Tampa. There's a part of town called Sulphur Springs because Sulphur Springs is there. And some of this work was done by Michael Garman, a former graduate student of mine, who was once a graduate student actually here in marine sciences. He moved over to biology and joined my lab partway through his graduate career. And Bobby Sharping, who's right over there, are the ones doing this. Now, Michael had mapped this conduit, and this is over a kilometer of passageway. And notice it goes right under Nebraska Avenue. So you go in here, and this is the Greyhound track. If you've been in that part of town, you would know. And there's a branch here, the Alaska Tunnel goes off to the right here, and the Orchid Tunnel goes further up that way. There are some sinks that were once connected, but they think those connections are no longer working. There's a spring pool here, and that spring pool back in the 50s and the 40s was a big attraction because there were no houses in that area. This was north of Tampa proper back then, and prior to air conditioning, this is where people would go to cool off in the hot summer days because the water was 72-degree spring water all year round, right? And so there were actually hotels and all kinds of things around it. And that existed until the 60s when there was a major fire and all the buildings burned down because they're all old wooden structures. When you go into the conduit, the depth changes. So when you first go in, it's at 10 meters, drops down to 25 meters, almost 30 meters. Then it kind of wobbles around, and then there's another sudden drop, and then it goes back up, and then there's another sudden drop. So we think that these three areas here where these sudden drops are represent kind of vertical developments or fissures or cracks in the rock formations. And you'll see that will be important as we go further into this. This is what it looks like today. So here's the concrete walls. The bottom is still sand. The entrance is about down here, or actually about down here under the water. This is the little gate we jump into. This is all the equipment it takes for Bobby and I to spend an hour down there. And this is the entrance. It's, the water's flowing. That rope, you're looking into it, so it's a horizontal entrance. And this rope is sticking straight out because of the high current. That's a 1/2-inch thick nylon rope. And it's sticking straight out like it's magic because the high current. So it's really hard to swim in. The entrance height of this is less than, it's probably 2.5 feet tall, maybe, so you kind of got to wiggle in. And you have to grab the rope to pull yourself in because the current is so high, you couldn't just, it'd be really hard to just swim in, especially with all the gear that we have to carry. So this is what it's like. Bobby took this video of me kind of bumbling in. So you can see where the entrance is, and you can see the rope there. And if you look around my feet, you can just see the sand. Look at it kind of roiling around even before my fins hit it. We'll let it go one more time. So it's a lot of effort to get in, but this is just showing how forceful that water is when it comes out through that restriction. Once you get a little further in, the circumference or the diameter of the conduit widens greatly, and so the water velocity slows down and you can just swim normally. So the main conduit's about 30 meters below ground. There's a thick brown biofilm on most of the surfaces, and I'll show you some of that. And then you'll find these little vents that pop into the main conduit, and around those smaller vents you'll find these white, lighter colored biofilms. And the vents bring in usually salty, sulfitic water. So there's something else going on with those other communities. But before we can really understand that biology, we have to work out the hydrology of the system, and that's what Bobby has spent most of the last 1.5 years figuring out. So the complication to all this is the city is pumping water from that discharge pool, and they only pump in the dry season. And the concern is the Hillsborough River during droughts will flow less, so there's more likelihood of Gulf saltwater coming up the Hillsborough River at high tide. So somebody, the state legislature decided that the city should be forced by law to pump water out of that pool, out of that spring, into the Hillsborough River to make up for the loss in normal Hillsborough River flows. So they call that maintaining the minimum flow of the Hillsborough River, and it's a state law that they have to do it, and they do it. And when they do, all kinds of crazy things happen. So that's what we'll be looking at. So this is part of what the homework Bobby did. So this is a map of the area. There's Sulphur Springs down here. Here's that Hillsborough River. And this is the green swamp area where the Hillsborough River comes from. He found that the Swanee limestone is exposed over here. The Tampa limestone is exposed here. And if you look at it from the side underground, here's the green swamp. Here's the Tampa limestone. Here's that Swanee limestone. So water will flow kind of downhill underground toward Sulphur Springs. And there's a well up here that we'll talk about a little bit later too. So what Bobby did is he looked at the historical data, and there's data going all the way back to 1942 on the salinity of the discharge, the amount of discharge and all that from that spring. And there's 2 dotted lines on here. This is when the city installed that pipe and started pumping, but they were pumping to add water to the drinking supply for Tampa. So it wasn't a ton of water, but they were pumping water out of there. So pumping started in 1963. But then 2002 is when they started that maintaining the level of the Hillsborough, minimum flows in the Hillsborough River. So this is when the pumping really started going crazy. And if you look at the trend before this diversion here, and you look at the data that's out there, you would make a line like this. So gradually increasing salinity from maybe 500 up to, maybe up to about 3,000. But when you look at the more recent data, he found a lot of scatter in there, but if you actually model a line to all this data, you get this kind of a line where the increase in salinity is really going up fast. So to look at it more closely, he just picked this part here, just the data in here, and that's going to be on the next slide. And I'm saying that because you won't see this trend line when you look at just this data, because it's too scattered to show a clean trend line. But in the next picture, you'll see that data. So here's the original figure, here's just that part of it, and greatly expanded with some other data thrown in. So I'm going to just kind of step you through this. So this is, they have measured since 2009 the level of water in the pool. And because we know that when they pump, the pool level immediately drops at least a meter, that tells us when the pump's running, right? So every time this blue line drops, that means the pump's running. Oh, it's not running, now it's running, not running, now it's running a lot. Here it's running a lot, right? Here it's running a lot. So we can look at how often they're pumping and when they're pumping. And this is the aquifer level at that well up at that exposed Swanee limestone that I showed you in the previous a few slides ago. And what Bobby expected was that the salinity, and this is the graph of the salinity, ignore the different colors, but just the way this line goes, that's the actual specific conductance as measured over this whole period of time. But what he found is the aquifer level itself only explained some of the data. It didn't explain all the data, all the salinity data. But when he looked at a combination of aquifer level and pool level, in other words, whether the pumping was going on or not, then he saw direct correlation. So here, there's no pumping going on, and the specific conductivity is kind of matching the aquifer level. Here they turn on the pumps, you get a spike in salinity. Every time they run the pipe, salinity goes and spikes. And then the pump was off, pump back on, pump off here. So what he was able to do was, by combining the pool level and the aquifer level into a correlation analysis, he got very high correlation to explain this trend here that he couldn't get by using just pool level or aquifer level by itself. But combining the two, he gets really highly significant correlations. And he also plotted a rainfall down here just to get an idea of kind of what the seasons were like. So this is data for over a 9-year period. So the weird thing that puzzled him for a long time was why the salinity in the spring increased in the wet season. We would always think that you have lots more water coming in, it's going to dilute any salt, and you're going to get less salinity in the spring. But it didn't work that way. So it could be partly explained by increased aquifer level, pressurizing maybe the deeper part of the aquifer that contains salt water and forcing that kind of up through the conduit. He also noted that the salinity increases in the dry season when pumping occurs. And he thinks that's because when you lower that pool level, that allows salt water from the Tampa Bay, sort of underneath the Tampa Bay, to come in, up in there and get into the conduit. And ideally, if no pumping were going on during the dry season, that is when we would expect the salinity in the conduit to drop. But they always pump in the dry season, so we don't see that. So again, here's a picture of what he looked at. So here's Sulphur Springs area. These are a bunch of wells that he brought in to look at to kind of explain this. And this Tourist Club well is the one nearest to Sulphur Springs. And that one turned out to be the key well. So let's go back to this map. So we have this conduit here. And just to remember the profile when you're in the conduit is there's these 3 vertical kind of developments in the cave that might be, they're probably fractures that go down further into the rock. And that's where the salty sulfidic water seems to come from. He looked at some of the biofilms. So this is a biofilm that you normally see around most of the conduit walls. It's brown. My former graduate student, Michael, had tested it. It's does iron metabolism, sulfate reduction. When we looked at it with a microscope, you get this it's a mixed culture of several different kinds of bacteria. And then these white biofilms around the salt vents, like here's Bobby collecting from near a salt vent. There you get what looks like almost a pure culture of stuff that looks very much like thiothrix, which is something we'd expect if the sulfur oxidation were occurring there. So that's the little tiny bit of biology we have so far. So here's Bobby's model of how it probably works. So again, here's the kind of cross-section, so the Tampa Bay out here, inland here, the green swamps out here, and the vertical is exaggerated. But here's the Swanee limestone, here's the Tampa limestone. The Swanee limestone has a confining layer right smack in the middle of it. I'm not sure why that came to be, but you can imagine if water lands on the Tampa limestone exposed here, it'll flow downhill underground, and that will feed in. And here's the spring, right? So here's the cave, and here's those vertical developments in it, right? And then you can imagine fresh water can come in the Swanee limestone under this confining layer and flow into this area as well. But think about these fractures, and whenever you have a path to the surface, like a well, you'll typically get this what they call upconing. So the mixed water is down here, mixed salt water is down here, and it's going to work its way up through these fractures, so you'll see this upconing. So the fractures draw up this saline water, and how much is controlled by the pool level here. In the dry season, when they're really actively pumping the spring pool, and they've been doing this for a long time, what we think is that water, this upconing is happening more often, and a lot of that salt water is sort of filling in areas in this Swanee limestone underneath the confining layer. It probably is just building up in here. And he knows that because that one well, what was the name of that well? The Tourist Club well is at that depth, and it has salt water in it. Other wells that are deeper or shallower in the area don't have salt water in them. So there's some good evidence for that. When you go pumping like crazy in the dry season, you're going to be extracting even more, and you're going to get more upconing, and this trapped salt water, or this sort of reservoir of salt water is going to get saltier and saltier. And then to explain wet season increases in salinity, in the wet season, you have water flowing and feeding the conduit here, but then the water that comes from that exposed Swanee limestone underneath this confining layer will come in, and it's going to get in this area, and that's going to push this water up into the conduit, causing the increase in salinity there. So it appears that the pumping from the sulfur springs to maintain minimum flows to prevent saltwater intrusion is actually causing saltwater intrusion, but in the spring as opposed to the river. So eventually the spring could become so salty that it couldn't be used. They already can't use it for drinking water. It's too salty for drinking water. But pretty soon they won't be able to use it for river flow. And the ongoing studies in my lab that Bobby's going to do is going to be to characterize the microbial communities. But he expects to see increase of those sulfur oxidizing communities that'll replace those older iron metabolizing sulfur reducing communities. And those sulfur oxidizers then produce sulfuric acid that enlarges the fractures, and thus we're going to get more and more salinity, more anoxia, and possibly sinkhole formation. So all the bad things that happen could happen. So not to be depressing or anything, but our next job is going to be to try to convince the city to go to the legislature and maybe stop the pumping, because the other reason they pump is it makes a nice warm water refuge for the manatees. So we haven't been able to dive in it for the last month because the city's afraid to shut off the water, because then the manatees would go away and then the public would get mad. So it's a very situation, a little sociology involved. So final story, Hospital Hole, Weeki Wachee River. This is work done by Madison Davis in my lab. And the diving, of course, is Bobby with me, and all the lab contributes to chemistry day. When we come home with all these samples, we get back at 7 or 8 at night, and there's 5 or 6 students that have to stay up all night analyzing all the samples, because I don't like them to sit around for a few days. I want them all done ASAP, so we get more reliability in our data. So this is a long ongoing project. It's going to be a PhD thesis, but I'm just going to show you the date for one collection date in March 2016. But we have been collecting data every 2 months since then. So we have a lot of data, the same data I'm going to show you now, but for over for bi-monthly for a year, and we're going to keep collecting that data for another probably year and a half. So what I'm going to show you is a static picture of what you see in this hole, but what Madison really wants to do is look at the dynamics of that over time. So that's going to happen. So the worst thing about working in the spring are the vicious manatees. So as you can see here, this manatee is charging me, and I'm starting to look a little nervous. And here, I mean, he's almost got me. So I'm looking panicked. I'm trying to get away from that manatee. And here, I'm waiting for this manatee to go attack one of those kayakers. So once he does that, then I can go swim over here and see that dark area? That's where the hole is. That's where the hospital hole that we're going to go into is. And then here, this manatee was trying to take two of us out at the same time. So, I mean, it's as bad as the leopard seals you hear about in Antarctica, only we have to deal with it right here in our home in our own backyard. So this is what the sinkhole kind of looks like, a really bad cartoon I drew with a mouse, so it's not artistic. So imagine the Weeki Wachee River is flowing overhead, and it's only a meter or two deep, depending where you are in it, but it's flowing pretty fast. And that's where the manatees, the man-eating manatees live. They hang up there. They'll drop down into here sometimes, but they don't like to get down in here. So they mostly hang out here and kind of sleep. They'll find a ledge and fall asleep. So you'll be decompressing, and you'll feel like somebody's looking at you. And you turn around, and there's this evil manatee just glaring at you. So when you sink down here, you can see the halocline as you go through it. And then we're in what Madison has called the hypoxic zone. And it's got salt water, but not as salty as down here. So partial salt water. This water is fed by this conduit. These arrows I show them going down, they should actually be going out in different directions here because it flows straight out here. But that's the origin of this layer that we call the hypoxic zone. Then you go through what we call the chemocline, and you see this cloud layer there because particulate matter comes down, sinks through the less salty hypoxic zone, but then it's not dense enough to go into the anoxic zone, which is saltier. So it collects right on that surface. So that helps us identify where that chemocline is. And really, it's just the bottom of the hypoxic zone where the cloud layer is. But it's right at that interface. And here's what the vent looks like. So when you're looking straight into the vent, this hole back here, it's about half a meter in diameter. So it can put out a lot of water. This is actually Bobby looking into it. So the vent is to the right here. When you get down to the anoxic zone, there's no light at all. The only light is what we carry with us. No light can get through this layer here at all. So we're and here a lot of sulfide, a huge amount of sulfide, higher salt in there, and no oxygen at all. And there's a debris mound. We don't actually know how deep the sinkhole actually is, but the debris mound is at about 40 meters. So when we take one of those HydroLab data sondes down attached to a diver, it'll measure all these parameters as you're going down. So here's depth, and here's dissolved oxygen, for example. So as you go down, plenty of oxygen up in the river. As soon as you drop in the anoxic zone, it's pretty low. When you get anywhere near that vent, you get a little bump in oxygen. That's why I call it the hypoxic zone as opposed to another anoxic zone. So we do occasionally measure oxygen in it. But then once you get below that, you're in that bottom anoxic layer, you get zero oxygen. Salinity, very low up in the river. As soon as you drop down through that first halocline, you're down. In this case, on this day, it was around 10 or so. Then when you get down into the anoxic layer in the very bottom, it goes up to about 15. pH around 8 in the river, down to about 7.4, and then when you get that anoxic zone, it drops down a little bit lower. And if you look at redox potential, you kind of expect this as you're going deeper and getting more anoxic. You expect that change in the redox, in the ORP. So these are what allowed us to come up with the layers here in the first place. But then we look at a lot of chemistry. So here are the layers. This is the surface, that's the water. This is the hypoxic zone. the chemocline, the anoxic zone. And this is data from one day. So we measured alkalinity, ammonia, nitrate, nitrite, phosphorus, sulfate, and sulfide. And these are replicates. We do lots of replicates so we can do statistics to compare it to the biology, which I'll show you in a minute. And she had some, you know, this was early on, so there were a lot of where students dropped a sample in the lab or something happened, you know. But still, we always had at least 3 replicates of each one. But look at the values. Look at sulfide, 30,000 micrograms per liter. That's a lot of sulfide. If we get any water in our mask when we're down there for more than about 15 minutes, our eyes start to burn. I mean, they burn like you know, like somebody's put red hot pokers in them, and you can't do anything about it because if you take off your mask, you're going to make it worse. So you have to wait till you get up in the less sulfitic area, and then you can rinse it out. But a lot of variation, but this is all just from one day. This is one day's worth of data, and we have this every 2 months for since then. So if you look at a principal component analysis of that chemistry data, and we combine the chemistry data from that chart I just showed you and the hydrolab data I showed you in a previous slide, what you see here is the blue here are the hypoxic area zone chemistry, and the replicates, we use 3 of each to do this chart. They typically are a little bit higher up on this scale. And then the red ones are the chemocline, and they tend to be a little bit lower. This one was up there, but these were lower. And then here's the anoxic zone, very, very different from the chemistry. So the chemistry of the hypoxic zone, chemistry of the anoxic zone, hypoxic zone, and chemocline. And notice that all 3 of these layers are statistically significantly different from each other. But there does seem to be, in this case, a little bit overlap between these 2, but overall we get a difference. So this is what the chemistry looks like. Now I'm going to go into the microbial analysis. So we used Illumina sequencing. We used 16S genes, the V4, V34 region. We had about 60,000 sequences per layer, but in different samples. So we did 3 replicates of each. So that way we could apply statistics to this data, because most people do a microbial community analysis and they show you a pie chart. And we wanted to go a little bit beyond that. So then we use that mother software to identify the operational taxonomic units. And then we look at similarity of communities by these operational taxonomy units, or OTUs, in each layer. And we limited the analysis to the 2,000 most abundant ones in each layer. We had 9,190 of them, but most of those were singletons, so we just took the top couple thousand for primer E analysis and Permanova software. And then Madison took on the functional analysis. So this is just 16S data. It doesn't really tell us what the bugs do. So what she did is she took the top 100 OTUs in each layer, blasted them to identify the nearest identified hit in GenBank, and then she would look those up and see what functions they had metabolically that were known for those. And we realize it's a provisional identification, usually to genus, but we can get some ideas about it, you know, when you look at the big picture. So she looked up known metabolic functions of them, and then she could compile a model of the metabolic functions in each layer's community. So here's sort of some of the data, and of course you don't want to see all our data, but this is the top 24 genera by sequencing as identified from OTUs that we sequenced. So notice there were 20,000 sequences that make up these top 24. And look, 15,000 of them, so 3/4 of them were this one genus, or at least provisionally identified as this one genus. Same thing here, 10,000 out of the 29,000 were this one genus, and here 12,000 out of the 22,000 were that. So the ones that are in bold are the ones that are predominantly found just in that one layer. So the ones in bold in this layer are the ones we found primarily in the hypoxic layer. The ones in bold here are the ones we found primarily in the chemocline, and the ones here are the ones we found primarily in that anoxic zone. So there's definitely different communities. You can just see it by looking at those bold words on there. So we did a principal components analysis of that. So now we're not doing a component analysis of the chemistry. So ignore this, this wheel here and these things. We'll come back to those later. But these triangles now represent the microbial communities as identified by 16S sequencing. So here are the hypoxic zone communities, the 3 replicates, and they're nicely clustered together. Here's the hypoxic zone. Here's the anoxic zone, the deeper ones, and they cluster together fairly nicely. But then the chemocline ones are nicely sort of arrayed in here between the two. So there's a distinct biological community here that you don't see here or here. So it's unique. And that's what we were expecting to find, was a unique community in the chemocline. And then if you look at the chemistry, so in primer E, you can overlay the chemistry, so increasing temperature and pH from this way to this direction, and all these other things were increasing from the left to from the right to the left. Temperature, pH increasing from the left to the right, and all these increasing right to the left. Just to remind you, so here again, we'll look at the biology. We have the hypoxic here, chemocline here, anoxic here, and then go to the chemistry. And in the chemistry, again, a little bit different. You know, here it looks more like the hypoxic zone and the chemocline are similar, and the anoxic zone is very different. But again, you go to the biology, and you see that chemocline has its own distinct community. So here's the function table she built, and these are some of the things she looked at. You know, are they aerobes, anaerobes, photosynthetic? Are they methane oxidizers, sulfur reducers, et cetera? So she looked at a lot of different things. And from that, she was able to kind of put together this cartoon of what's going on inside there based upon her analysis of function from just the 16S sequencing data. So what you see is in the hypoxic zone, that's the only area we found photosynthetic bacteria. We found some sulfur metabolism there, a lot of nitrate metabolism. And one route that we found only in the hypoxic zone was this ammonia to nitrite pathway. It was the only bacteria that were capable of that, at least from the literature that we found only in the hypoxic zone. You go down to the chemocline, you see a lot more different kinds of sulfur metabolism going on here. And you know, these are all attached to carbon, use of organic carbon, so that's what these represent here. But lots of different sulfur metabolism going on, lots of nitrate and nitrogen metabolism here. And again, we find a distinct thing, nitrate going to nitrite. We only found that in this chemocline layer. Then we get to the anoxic layer, similar kind of thing, only here sulfide is really high, so she's got that in orange. And the only place she found bacteria that at least from the literature, can take sulfide to sulfite was down there. That's why this is in orange. There's no oxygen here. Again, these are carbon pathways in these little things. And here's the nitrogen. And here she found the ammonia to nitrogen, which is the Ammonox reaction. She only found that down here. And she only found, I think, the methane oxidation down here. But if you find methane oxidation, what else do you expect to see? If something's oxidizing methane, where did that methane come from? Anybody know?
Decomposition.
But how? You want to answer that, Bobby?
Methanogenesis.
Yes. So there must be methanogens down there. But methanogens are archaea, not bacteria. Our primers supposedly would do both, but we found like 10 archaeal sequences in our data, so we've already switched primers. But yeah, if there's methane oxidation going on here, there has to be methanogenesis going on as well. And we just couldn't find those. So that's kind of what we saw. This is like a vignette of one day in Hospital Hole, a lot going on in there. We hope eventually to do direct metagenome sequencing, look at actually what the enzymes are and all that. But for now, this is a relatively inexpensive way for us to work and get some ideas. We've done this before for some other projects I didn't have time to talk about. So for Hospital Hole, the first thing that's interesting that I didn't really talk about, but I want to talk about now, is that because there's a river flowing overhead, you don't get seasonal changes like you would expect in a sinkhole that didn't have a river flowing over it. So imagine a lake. In a lake in the winter, when that cold winter wind blows through here in Florida, it chills the surface layer. And even though it may be less salty than the water below, if it's cold enough, it'll sink down through that anyway and displace some of that deeper water. But because we've got the Weeki Wachee River flowing over and its spring temperature, it's always 72 degrees, that's why those evil manatees are there in the winter. That sort of insulates this system from normal turnover type events that you would normally expect to see, which is kind of interesting. So that makes it unique compared to any other sinkhole we've ever studied. Each layer there seems to be geochemically and biologically distinct. The hypoxic zone originated from this shallow coastal mixing zone water that comes in through that big spring or vent down there. I didn't know that. I've dove Hospital Hole for almost 20 years. I never realized that water in the middle, I've always assumed it was just Weeki Wachee River water that had kind of sunk down there. But it's not. It's coming from a separate source. And we found that out from all our analysis. Then that anoxic zone at the very bottom, that's also obviously mixing zone water, but it's coming from deeper in the coastal mixing zone, probably, and accounting for its higher salinity and the higher sulfate that we find in there. And what's really nice about this is I asked Madison to look at microbial communities studied using 16S in the Florida aquifer, you know, for us to compare our data to. And guess how many papers she found on that? She found 0. Not a single paper on the bacterial communities in the aquifer based on DNA sequencing. It's all based on, well, they see sulfides, so there must be sulfate reduction kind of stuff, you know, or we see nitrite, therefore, you know. But nobody has actually looked and used modern methods to see. So what we're kind of hoping is that the communities in Hospital Hole could represent the same communities present in the coastal aquifer. So what she's going to do now is go to SwiftMud and get them, she selected some wells that SwiftMud monitors, and get them to let her sample in some of those wells and compare what she sees in Hospital Hole to what she sees in the aquifer. But this at least gives us an entry position into looking at what microbial communities are doing down in the aquifer. So that's the ultimate goal, is to figure out what changes she might see over time and what's driving those changes, because that's the theme in our lab, what changes microbial communities in these systems. So Vicki in the lab, she's working on carbonate dissolution in another story that I didn't have time to talk about. Madison's looking at using salt-tolerant microbial communities for fish waste digestion. It turns out if you take, like if you're doing aquaculture on drum, it's a salt fish, you can't take drum poop and just throw it in the sewage system because it's so salty it kills off the microbes in the sewage treatment plants. So the idea is find salt-tolerant ones for that. And we think that the more systems we study, we'll see a little bit more about the changing role the aquifer plays, especially in a rapidly developing area like the Tampa Bay area. These are all my current graduate students, former graduate students, collaborators, undergraduates. I bolded Vicki not because she's here, but because as an undergraduate, she actually acts more like a graduate student in our lab. In fact, she's starting a PhD program at University of Tennessee in Knoxville to work on similar stuff. So that's kind of nice. And these are the myriad of undergrads over the last probably 5 or 6 years that have pulled all those all-night chemistry stints. So I have to thank them. So with that, I'm ready for any questions if you have any.
I didn't see Lynn Vasher's name there. Is he still?
He's still around. I see him every now and again. He's more into education stuff, I think, right now. STEM ed stuff. But you do. It's kind of marine. Got it. Yes.
It seemed like, for some of the data there in Hospital Hole, looking at it from back here, that there's species that are in your hypoxic zone and in the anoxic zone that aren't in that,
Chemocline.
chemocline.
Mm-hmm.
So, is there any idea, like, I guess that's not acting as any sort of barrier for them to move up and down in the water, or is it that there's just species down there in a population that's already set that?
We don't really know, but I could imagine that there's going to be connectivity between them through the chemocline. For example, what if a particle is going down through the water column that's heavy enough to break through the chemocline into the anoxic zone? If some bacteria in the hypoxic zone adhered to that particle and it made it through the chemocline, it would. So there's got to be a certain amount of connectivity. I think we'll be able to find out more about that when we're able to analyze multiple series of these dates. Remember, this is just the first part of probably a 2-year study. So I don't know. I don't know the answer to that, but it's a good question.
Which blue hole is that in the Bahamas?
It's a little tiny blue hole. I forget. Bobby, do you remember this one?
That's Watley's hole.
Watling's hole.
Say again?
Watley's.
Yeah, Watley, that's the wall around that's where they used to raise sea turtles. Anyway, yeah, that's on San Salvador.
Yeah, it's
Which island is it on?
San Salvador.
Oh,
So somebody a long time ago built a rock wall around here because they were trying to raise sea turtles in it for, I guess, food. And they abandoned it. But we did a really neat study there that it's working its way through the literature. Yeah, through getting into literature, yes.
In your anoxic zone, you had E. coli. And
Yeah.
Which it triggered my interest because I wouldn't expect that to be in an anoxic zone, especially in that. Do you think it could be from like fish that's fallen that are decomposing from the?
It could be lots of things, but oddly enough, we find E. coli almost everywhere.
it's a dark sucker, that's
You know, there's a site we're studying in Sicily, and we find E. coli is the primary component of this really thick snotite stuff that's
Yeah.
down in there.
Their biofilms are really intense.
Yeah.
Is that one next to a highly populated area where it would be a human-related one, or?
Probably cattle
Yeah.
above. This one, there's a lot of people around here. You know, it's in Weeki Wachee Gardens,
Yeah.
and all those houses, I'm sure, are on septic tanks.
Okay.
I would guarantee you they're all on septic tanks. And, you know, they've cut canals. So the Weeki Wachee River goes through, and they've cut all these canals. And these are really old. This is kind of the ghetto part of the Weeki Wachee area. So there's like rundown mobile homes on these lots, and, you know, just sort of decaying old houses that have been there for, you know, maybe 30, 40 years.
Yeah, maybe look up to see if E. coli was anoxic, because I never.
Oh, it can be anoxic.
Yeah,
Yeah, absolutely.
I Googled it.
Yeah. So we have a guy in our department that studies E. coli, and we were puzzling about that snotite biofilm.
Yeah,
So we went to him, and he got all excited about it. Because,
yeah, that's the first thing I tell all my micro students is that it makes an intense biofilm. It's very noticeable.
Yeah.
So I'm not surprised.
yeah, it's a great thing, but it's a major component in here.
It's awesome.
Yes?
Do you notice any similarities between the different holes in regards to which species form which biofilms, or is each hole almost kind of like its entire separate assemblage?
We have noticed a certain kind of biofilm in Hospital Hole at the bottom that we have also seen in another sinkhole called Hudson Grotto. And we've also seen it in an offshore sinkhole called Jewfish Sink that we've studied quite a bit. And so a side project in our lab is we've collected biofilm from all three of the same appearing looking biofilm from all three locations. And we're going to do the sequencing on all those samples. We've also got the water chemistry for the water they were collected from as well. And I think it's going to make a nice little story because they look similar, and I've always thought, gee, maybe they are similar, so we'll find out. I have no idea. But they're very distinctive. But you'll find in one hole, you'll find like 5 or 6 different kinds of biofilms, depending on where you look, or more. So that's, and there are so many of them. I mean, you could spend your entire life just analyzing different biofilms. And I don't know how important it is, but boy, you know, if you see something that's showing a trend like what Bobby noticed, the sulfur oxidizing bacteria containing biofilms overgrowing what used to be iron metabolizing and sulfur reducing biofilms, then it may become more important. But you know, you could just start swabbing around this room every day and do the 16S analysis and look at it every 2 months, and you'd find all these differences, and you could, I mean, you know, you could. The question is, you know, where, who cares, and what's important and what isn't. And we just don't know at this point. You know, I mean, people are doing microbiomes every possible place they can. Yes?
Okay, so I don't remember the name of the location, I'm sorry. The first story, when you saw the...
Double keyhole.
Yeah, so following the tropical storm, when you saw the general, like, levels increase and kind of stay increased,
Mm-hmm.
I guess the question is, A, is that pretty common throughout the aquifer in Florida, and B, how does that time scale kind of compare to the general draining of
So,
the aquifer?
it kind of depends on the spring. So it depends where the recharge area is. So if the recharge area is close, you get a heavy rainfall, you might see an almost immediate increase in the discharge. And that's what seemed to happen at Double Keyhole. But Bobby's spring, you know, Sulphur Springs, there's a longer delay between a rain event and increased flow there. And that's because the recharge area is all the way out in the green swamp. So that water, it takes it a while to get there. So
So are they pumping for drinking water from that area? I know they're pumping to flush the rivers out.
they no longer use that water for drinking water. It's too salty.
Right, but but originally was.
Originally it was, yeah. Mhm. It was part of the city of Tampa's water supply. And as it got saltier, they decided that they couldn't use it anymore. I guess there's limits. You know, it doesn't taste that salty.
Cohout circulation, Fran Cohout did that work years ago, showed salt water comes in under the, well, intrusion.
Yeah.
Just about everywhere in the state of Florida.
It does. You go deep enough, you're going to run into salt water anywhere. Yeah. Yes?
So I wonder, do you see hypoxia or anoxia for all the springs you observe, or just versus springs?
Well, I know that a lot of springs I've dove in this area have oxygen in them. It depends. So, for example, Eagle's Nest has been in the news a lot lately because a couple more, a diver died in there recently. And it's a very deep sinkhole that leads into a very deep cave system. And there you see stegobitic crustaceans, albino crayfish and things in there. So it just depends. And I'm not sure what I think it has something to do with microbial activity. I think if you have a lot of microbial activity, they might deplete the oxygen. I'm not really sure. Maybe Bobby knows.
Yeah, tied into that too, I guess it's a question of residence time. If the spring is getting recharged really quickly, then even if there are a lot of microbes, they'll have a lot of time to heat up all that oxygen. Compared to if it's percolating not through a conduit, but just through the groundwater matrix, you know, a lot more slowly, and so they have a lot more surface area for microbes to even heat up that microbial oxygen too. So.
They metabolize it. Yeah.
I have another question regarding the pumping. So my question is, instead of like totally shutting down the pumping, is there any like quantification or model to calculate the optimal number we want to achieve instead of just shut down the pumping? Because I see the Hillsborough River discharge flushing from your, so which means they don't need the same amount of water pump from the spring to the main river.
Right.
So is there any way to quantify or to model?
They have big, thick documents like this about it, and they have modeled it to death. Somebody has made a lot of money writing those reports, I suppose. But when you look at it from a more simple point of view, it just doesn't make any sense to me. But that's going to be our thing to do next, I think, is to try to convince people that maybe they don't need to pump, or maybe it's not going to help in the long run. It may help. temporarily. But it's great. So after they've been pumping, you know, I was talking about how the flow comes out so you have to drag yourself in on that rope to get into that entrance. They've been pumping and they shut off the pumps for us. You just swim right in. Because there's no water flowing out, or if the water's flowing out, it's just barely flowing out compared to normal. So it's really bizarre. So we'll go there. The pond level will be down 2 meters or so below where it normally is. So they'll shut off the pumps and we'll be getting our gear on. And by the time we got our gear on, the pond is up to its normal level. And we jump in and we go down there and there's no water flowing out. It's just because they've sucked it all out. I guess it takes it a while to recover its flow. So, and you know, we've been in there for, you know, over an hour and not noticed any more flow when we came out. So it's weird. Yes.
Sort of a follow-up to that, just out of curiosity. I know you said there's seasonal salinity changes, but do you think if the pumping stopped completely, it would ever freshen back to like previous levels? Or is that kind of gone?
Once you've got salt water in there, it's really hard to get it out. We were talking the other day, what was the term we were using?
Rinsing.
Rinsing, yeah. So there's the rinsing hypothesis about how long it would take to flush out salt water that's intruded. And it can be very complicated. But every time the tide goes up and down, that causes more mixing, right? When the tide's pushing water up and down through rocks, through all the pores, it's forcing a mixing to happen even more, so then it diffuses out even further. So yeah, I mean, if they stopped it, you'd still see an increase in salinity probably for a long time, if not forever, but it would probably slow down immensely, I would guess.
Okay, well, let's thank Dr. Garey again. And I do believe we have a TGIF celebration. Is that next week? Yeah. Oh, sorry. I apologize. I just saw the email just before I came down, so.